I direct a group at the MIT Media Lab and McGovern Institute, which is trying to develop ways to solve the brain. And what I mean by that is, can we understand how the brain works, and can we repair brain diseases? I think part of the problem of why this is so difficult is the brain is very complicated, and yet it mediates our thoughts and feelings, it confers upon us our identities, it makes us who we are. And so I can't think of a more important, in terms of pressing health needs, as well as sort of deep philosophical yearnings, topic to confront. It's very helpful to think about why it's so difficult to understand the brain. And I think there are two issues. One is space, and the other is time. If we think about space, the brain presents an extraordinary challenge. We have to deal with spatial scales that go from the very large, brain cells are the biggest cells in the human body by far, to the very small. The individual connections between brain cells, called synapses, are nanoscale in dimension, and they are coordinated with nanoscale precision. So you have these elaborate neural networks, wires that connect in elaborate 3D patterns. And within a cubic millimeter of your brain, you have about 100,000 of these brain cells connected by perhaps 1 billion connections. And those connections are jam-packed full of biomolecules, which are themselves nanoscale in configuration and organized with nanoscale precision. There are, 30,000 genes in the human genome. Each of those is encoding for biomolecules that can be arranged in complex patterns and that make these brain cells do what they do. So space, how do you cross all those spatial scales? That's an open challenge. And the other challenge is time. So if you care about memory, or learning a language, or how to ride a bicycle, or if you care about diseases like Alzheimer's disease or Parkinson's disease, these are processes that take years, even decades, to occur. And yet brain cells compute with what are, in a biological sense, extremely fast events. Within brain cells, you have electrical pulses that last about 1,000th of a second, faster than almost any other biological process that we study at the cellular level. And then these brain cells communicate with chemical exchanges that are also very brief, around 1,000th of a second. So how do you cross that gap from these very small quantum building blocks to something like Alzheimer's or recalling a favorite song from childhood? So in my group, we try to build tools to help biologists, and in particular neuroscientists, to cross space and across time. And the hope is that we give these tools away freely, we can empower people to really study how the brain works and how it goes wrong. And that latter point is very important. This is not just about philosophy, as important as that is. Can we understand what it means to be human, to have thoughts, to have feelings? Can we also solve brain diseases, which affect a lot of people? And if you look at the names here on this slide, these are some of the most common or most severe conditions, which have a heavy factor of problems in the brain. Basically, none of these can be cured. The treatments are fairly partial, and they often treat symptoms rather than core effects. Alzheimer's disease, for example, my own grandmother has Alzheimer's, and it's devastating that we can't do anything about it. And all of us, of course, if we live long enough, will encounter ourselves or probably have a loved one who will suffer from some kind of brain condition. Because of the complexity of the brain, there's not a lot we can do. So if you look at the numbers, they're a little bit disappointing. Developing a new treatment and getting it out of the lab and into the marketplace to help people takes almost a decade. The failure rate is very, very high, disturbingly close to 100% to actually get anything that does anything at all. And the cost is enormous. I think this number is actually a bit out of date. I think more recent estimates suggest about $2 billion US to develop a treatment from beginning until end. And to top it off, as I mentioned, these aren't cures. They offer to treating just symptoms, and the effects are partial, and pretty much all the treatments have some kind of side effects as well. So I'll tell you two short stories today, one about space and one about time. And I hope to convey how we can develop very fundamental technologies that let us look at and control what goes on in the brain. And then from those technologies, we give them to thousands of groups all over the world to make discoveries. And from those are starting to emerge actual diagnostics and therapeutics that are now actually entering human trials. So let's start with space. How do you understand how a brain network is made out of brain cells, and how a brain cell is itself a network of biomolecules, these 30,000 odd, now much more we know, biomolecules that are encoded for by the genome? Well, I think we've all seen pictures that look like this, brain scans, right? They're popular because they're non-invasive. You can use these with normal human volunteers. Perhaps 500,000 studies have been published using brain scans. But the dirty secret is these blobs, or voxels as they're called, are actually very, very large. They contain millions and millions of cells. And we know that two cells that are next to each other can do completely different things. At the other extreme are microscopes. So with microscopes, you can see very tiny things. This is how brain cells were found in the first place over 100 years ago. But even these cannot see the tiny nanoscale building blocks of life that we need to be able to map if we really want to understand how a brain network operates. So in my group, we started thinking about how could you actually get finer resolution than you can get with a microscope. And so we started thinking about chemistry. Turns out there's a whole class of materials, such as the active ingredient in baby diapers. And what these materials do is they can swell enormously. So we started thinking, if everybody else is zooming in, why can't we blow the brain up? So people studied these responsive polymers, or smart gels, they go by many names. You add water to these polymers, and osmosis draws the water in, and you get this swelling. And these are very highly charged polymers, so like charges repel, they blow up, and you get these 1,000-fold volumetric increases very, very rapidly, in a matter of seconds to minutes. So we started thinking, what if instead of just taking a piece of baby diaper and just having it sit there, or try to stick it into the brain, what if you could chemically weave these threads of swellable polymers so densely and so evenly that, like a special kind of spider web, they would wind their way around the biomolecules in between them, penetrating the tissues so that they would form this very even web. And then if we did it just right, maybe we could blow the brain up. So we had to invent a couple chemicals to make this work. In this cartoon, the biomolecules are shown in brown, and we had to invent anchors, little molecules that would bind and attach a little handle to each of the biomolecules, shown in this cartoon in purple. So if every biomolecule, every building block of life has a handle, maybe you could pull them apart. The second thing is to weave the polymeric threads. Can you make a spider web that interdigitates all the way through the specimen, around and between the biomolecules? And it turned out in 1981, a group at the Max Planck Institute figured out how to do that. You can soak a preserved piece of tissue in a solution, and these monomers, as they're called, the building blocks, will form these long chains. And when the chains encounter the handle, they'll attach. So if you think about it, we have the polymers that can make force and the handles that can convey the force. Isn't that what we need? But there's one last thing we have to do. The biomolecules are perfectly happy where they are. They don't like being separated. And so we have to loosen everything up. We use detergents, for example, or we apply heat. And we can get them to lose their allegiance to each other so that when we add the water, the polymers will expand, but this time, the building blocks of life will come along for the ride. They're going to expand away from each other. So the idea here is, in contrast to the first 300 years of biological microscopy, where people zoomed in using little pieces of glass to make lenses, can we blow things up so that, sort of like a constellation of stars in the sky, we have all the biomolecules with the correct relative organization, but far enough apart that you can image them with cheap optics. And in fact, we are even talking with people now about using cheap microscopes built on modified cell phone cameras. Imagine blowing up a bacterium or a virus, and you take a picture using a ubiquitous imaging device. Could you bring diagnostics into the realm of the very inexpensive? So here's a little time-lapse movie of a piece of mouse brain tissue. People often use mice in neuroscience, and we polymerized it earlier. And I hope what you can see now is we just added the water, and this brain or piece of brain is growing before your very eyes. So it's probably only a matter until some Hollywood scriptwriter makes a horror movie about this. Okay, so what we can do with this? Well, we can actually start to make maps of the brain. We can take a brain, this is a part of the brain involved with memory formation, and blow it up until it's 100 times, 10,000 times bigger in volume. And so now you can see those individual biomolecules. You can look at their organization with the spatial precision that you need. But you can do it across extended spatial scales, because you can use cheap devices, like inexpensive microscopes, to see these extended objects. And so we're working now with distributing the technology to hundreds and hundreds of different users to look at brain maps. Can we actually identify the connections between brain cells and read out what a memory actually is? Could you look at the organization of a brain circuit with such precision that you could simulate an actual decision or a feeling in some kind of computer software, for example? And as importantly, can you map out where diseases begin? Could you figure out where the beginnings of Parkinson's disease or epilepsy or Alzheimer's disease actually start in the brain? Because if you can catch these diseases earlier, maybe you could pinpoint how to treat them better. And that's an important theme here. One of the fun things about brain science is that we're climbing this very tall mountain, right? It's going to take decades to solve the brain. And along the way, though, because the brain is so complicated, we find that our technologies can help a lot of other people solve biomedical problems. And I'll give you an example from the realm of cancer. So after we published our first discovery that we could evenly expand a biological system, a preserved one, of course, not a living one, a lot of doctors and pathologists started calling us up and said, wait, many diseases, in fact, most diseases in many countries, if you can detect them earlier, you could cure people better. But it's really hard to detect a disease early because the changes are very, very small, right? How do you see something when it's early? The changes are almost invisible. So one of our collaborators is an expert in breast cancer and suggested that we try to do early breast cancer diagnosis. Early in breast cancer, doctors will disagree half the time about the diagnosis, which is not good. So we expanded actual breast cancer biopsies collected by some of our collaborators, and we found that we could use these expanded specimens to train a machine learning algorithm, an AI, if you will, to do much better early cancer diagnosis than before. And so we actually spun out a company called Expansion Technologies, where we're now designing large-scale clinical trials to see if we can actually detect diseases far earlier than before, which could help save lots of lives. So that was the story about space. And now I want to tell you a story about time. The high-speed temporal dynamics, these electrical pulses, these chemical exchanges, are daunting to observe and to control. And so one of the quick questions is, could you do it very precisely? If you could control brain activity super precisely, you could try to test out a pattern of brain stimulation. Oh, look, here's a pattern that actually could cancel out an epileptic seizure or help somebody with Alzheimer's disease. So my co-inventor, Carl Dijsselroth, and I, when we were both students, we tried to think about basically every way that we could control a neuron's electrical pulses, a brain cell's electrical pulses. And so we just started going through all the laws of physics, sound and mechanical and light. And we decided light would be great if you could do it, because light, of course, is as fast as anything ever gets, and you can aim it really precisely, right? So then, how do you make brain cells sensitive to light? Most of them aren't. You need the equivalent of a solar panel that you can somehow stick into the brain cell, right? Then you have to get light in. That's not so hard. The brain doesn't feel pain in the conventional sense. Even human patients have electrical probes put into their brains while they are awake. So we could put an optical fiber into the brain of, let's say, a mouse or another animal common in neuroscience. Then the question is, how do you make those darn solar panels? So it turns out that the natural world, we just lucked out as pure serendipity, has evolved these for us. So in single-celled green algae, like in this little animation, there's an eye spot in the algae that if you zoom in, you can actually find little solar panel-like devices. And this algae uses this light sensor to help them swim around in bodies of water. So we just zoomed into the eye spot, and they're little ion channels, as they're called, pores, where when you hit them with light, such as blue light, they'll open up a little pore and let charged particles, ions, go back and forth across the membrane or the boundary of the eye spot. So that's very lucky, right? That's exactly what we wanted, except we didn't have to spend 50 years in making it. It's just sitting there in pond scum. So then we got even luckier. It turns out this is a protein. It's encoded by a little gene. And we can take the gene and use all these tricks from fields like gene therapy and so forth, which are increasingly widespread, and take the gene and put it into one of these gene delivery vectors and put it into a brain cell. And so then that was the big question next is, are we going to be lucky a second time? Will it actually work, right? You can't just take a plant gene, like from an algae, and jam it into a brain cell. Maybe it wouldn't work. And to our surprise, it did. We were able to take the gene and put it in with a gene therapy vector into a brain cell, and the brain cells would install these little solar panels right on the membrane where they should be. That was sheer dumb luck. And then when we shine light on it, that was even more surprising. These molecules were fast enough and strong enough and safe enough that even in delicate and fragile and high-speed operating neurons, they would work. With light, we could drive electrical pulses in these brain cells that look just like the kinds that are happening now in your auditory systems, as I say these very words. So how do you use such a technology? Well, again, there's lots of tricks from gene therapy that are beyond the scope of this lecture, but you can deliver the genes so that only some of the cells of the brain will turn on the gene, and some will not. So for example, there are little star-shaped cells in the brain called basket cells because of their shape. And one of our colleagues, Li Wei Sai, for example, has been using viral vectors to make just those cells become light sensitive. Now, why is that interesting? These cells actually undergo a lot of trouble in different neurological and psychiatric conditions. So for example, in patients with schizophrenia, these are the cells that are having trouble. And now you could shine light on these and try to jazz up their activity, right? Maybe you could repair the brain. Now, Li Wei is an expert in Alzheimer's disease, and so her group, using our technology, started to try to figure out, in mice that are engineered to have some of the genes that in humans can cause Alzheimer's, so mouse models of Alzheimer's, could you drive the brain with a pattern that could help repair an Alzheimer's state? So they put the gene in, they shined a light from a laser down an optical fiber, and they would drive the brain, and they found that if you drove the brain at a special frequency, 40 times a second, the brain's immune system would turn on, and you'd actually get cleanup of all this molecular debris, the amyloid plaques and the other problems that accumulate in Alzheimer's, which is pretty shocking, right? A pattern of brain activity confronting, one of the least treatable of all diseases. So then the teams went on and decided, could you do it non-invasively, right? I mean, gene therapy and optical implants, not anybody's idea of a good time, right? So the teams went on to try to figure out, could you replicate those patterns of activity non-invasively? No gene therapy, no light sensors from algae, no optical fiber, but could you do it through watching a movie, through your eyes or through your ears? And amazingly, in the study, they actually found that you could actually find movies that you could watch, these are mice still, and indeed, the brain's immune system would turn on and you get cleanup of these molecular problems of Alzheimer's disease. So Liwei and I have actually co-founded a company called Cognito Therapeutics, where we are now beginning human trials of effectively movies to treat Alzheimer's. And so lots of things don't translate from mice to humans, but it's a freaking movie. We should give it a try, right? Even if it fails, it's one of the cheapest failures in Alzheimer's history. So lots of people can try to use this technology for other things as well. Could you study addiction? Could you study learning? You can turn on neurons and figure out what they trigger. Do they initiate a healthy state? Do they initiate a pathological state? And so Chris Fiorillo is an expert on a molecule called dopamine. Dopamine is often called by the popular press like the pleasure center of the brain, but as we all know, everything's more complicated in biology than what you hear. And so he used a gene therapy trick to make dopamine neurons in the mouse brain, deep in the brain, sensitive to light. And his question was then, what if you activate these? What do they really do? Do they drive learning? Do they drive addiction? And so he came up with a very simple behavioral task. When mice go into one little spot in a box and poke their nose into a sensor, they'll get a flash of blue light. And if they go to another point in the box and poke their nose into a sensor, then nothing will happen. And here's what he found. The mouse goes, pokes his nose, gets a pulse of blue light. You can see it coursing down the optical fiber there, does it again. I think you're getting the pattern here. So basically, the mouse is working for light. Activating these cells is enough to make the brain do more of what it was just doing. And so this allows you really to start pinpointing where in the brain changes such as those that occur in addiction really are. Now you can go look at these cells and say, what kinds of drug targets could we try to find in these cells so that better drugs could be built? Or could we come up with non-invasive ways to modulate these cells? Kind of like the movies for Alzheimer's trick. Are there ways to actually cause these cells to do the right thing without a drug or without an implant? Flash forward to the current day, we're finding that molecules all over the tree of life can have powerful effects on brain cells. I'll just give you one example, which is I've told you so far about turning neurons on with blue light. Can we turn neurons off? And so it turns out that you can. We found molecules from many species of funguses and bacteria and archaea, which are kind of like bacteria but not the same, where they actually do the opposite. They'll bring negative charge into the cell and turn it off. So why is that helpful? Well, if you put the gene in that encodes for one of these proteins and shine light, you can turn off a cell. So imagine you can shut down the tremor in Parkinson's, or turn off a seizure in epilepsy, or try to delete a part of the brain and figure out, is it important for memory? And I'll give you an example from Akihiro Yamanaka's group, where they actually used this technology that we developed to try to understand narcolepsy. So in patients with narcolepsy, people fall asleep at random and inopportune times, and it's very hard to treat. And it was found that there's a certain cluster of cells deep in the brain that actually atrophies or dies off in patients with narcolepsy. So the next question is, well, these cells are gone. You can't build a drug that binds to those cells. You have to figure out what these cells do to the rest of the brain, right? If you did, you could try to replicate the function of these cells. So what Akihiro did was to engineer a mouse that you could turn off these cells, because they have our molecule in them, and they put a fiber with a laser attached to the brain so you can aim and turn these cells off. And here's what they found. So on the vertical axis is the number of these mice that are awake, and then over time you can see the orange bar, which is when they turn on the laser, and bam, they all pass out, right? Within half a minute, they're all asleep. No like error bars or anything. They're all completely zonked out here. And then you flip the laser off, and they wake back up. But you can see there's a bit of grogginess, and they eventually pass out again. So now you can really start to tease apart in a temporal way what these cells do and try to figure out, look, these cells might project to many parts of the brain. Can we figure out which one would be the one to treat with a new pharmacological agent? So going forward, where do we hope to end up? Well, one of my hopes is to try to actually combine these different tools, the temporal part and the spatial part, so we can actually make models of how the brain works. Could you understand how a decision occurs or how an emotion takes place? And of course, this sort of invent, discover, design, deploy pipeline, where I told you about two of the inventions, which actually now have even left the group and entered startup companies, one on early cancer detection and one on hopefully treating Alzheimer's disease. Can we actually expand how we do this? Can we actually come up with new models of innovation that can apply to really difficult arenas like neurotechnology? So I'm out of time. I'm going to end with this slide, which is far too long to go through all the people, but this is a very large, collaborative, you could almost call it omnidisciplinary effort. And so I think I can end there, and thank you for your time.
Thank you, Ed.
Thank you.
This was great. Thank you. Give me the clicker. Isn't that amazing? I mean, this is tool making, but tool making for the brain. Since you know more about the brain than almost any of us here, is there such a thing, it's a philosophical question, is there such a thing as free will? Or is it all biochemical processes?
Well, it's a good question. I mean, as neuroscientists, we try to figure out how much of our behavior and our thoughts and our feelings can be attributed to mechanisms in the brain. And it's interesting, some psychologists using brain scanning with humans have actually found patterns of brain activity that can predict what live human volunteers will decide to do later. So maybe it's both. Maybe there's free will, but it's also got a biochemical linkage, right? And maybe there's a point in the future where we'll understand why we do what we do. But who knows, right? This is the wild and crazy frontier.
But is there a hypothesis you have personally that there's no free will? It's just a myth. People talk about free will, but it really is a cascade of things that you can go back, hours, days, maybe years back until then a thought triggers or an emotion triggers.
Well, I think the big question that is sort of related to this is what is consciousness, right? So if free will is what you consciously decide, then we have to figure out what consciousness is. And the biggest problem in philosophy, in my opinion, is, well, how do we actually even understand what consciousness is? I mean, you don't know for sure if I'm conscious, right? Maybe I'm just a very, very accurate robot that you invited to come speak here, while my actual self is off on a beach somewhere. And that's actually not true. It's me. But the point
that
I
would
make
a robot say too. A smart robot would.
I was programmed
say
very
that
well.
too. Okay. Yeah.
But the point is that we don't have a consciousness meter, right? And so unless we have the ability to measure consciousness, how do we know for sure what causes free will? Mm-hmm. So I think that's kind of the big problem. And one of the things we're doing, although I didn't have time to talk about today, is we are trying to work on ways to stimulate the human brain non-invasively. Could you try to trigger a pattern of activity in the human brain? And what does that feel like? Could you trigger a decision? And what does it feel like when you are making a decision? Mm-hmm. It happens so fleetingly that it's very hard to pin it down. And yet if we could drive a decision in a laboratory environment, maybe we could actually try to understand what a decision mm-hmm. actually is.
Mm-hmm. If you could read people's thoughts, or even the subconscious, mm-hmm. would you do it?
Well, people are doing such things. So there are actually studies now by people like Jack Gallant at Berkeley where they'll image people in this brain scanner while they're thinking of an image, and they can actually guess what they're seeing, if it's a movie, or people are also looking at imagery. So that's been going on for about 10 years now. Mm-hmm. Mm-hmm. I think the big question is, what are we going to do about it, and can it go wrong? And looking at the CRISPR baby examples and others, is it time to have a global neuroethics consortium where we bring together companies? I think the largest neurotechnology group anywhere on Earth might be at Facebook, I heard. Where they have 80 people working on mm-hmm. brain scanners, I heard. Okay. Anyway, it might be the time now where big companies are getting into brain technology to bring together, lawyers, scientists, religious leaders, clinicians, big companies, funders, and let's decide what we want to do and what we don't want to do as a community mm-hmm. before somebody else does it for us.
Yep. Yeah. You talked about Alzheimer's and all the other brain diseases that we have and that we basically cannot solve mm-hmm. today. But it's a noble goal, of course, to solve these things. How about the other side of the coin of upgrading humans to have better brains, better thinking processes, like fewer thinking errors? How do you stand on that scale? What's your thinking on that?
Sure. So for me, it's all about the risk-reward ratio. I think there's a lot of, especially in the popular press, people talk about upgrading your brain with an implant. But implants, of course, have to displace brain tissue. They can lead to infection. They might not be easily reversible. I don't think we're going to see those used for pure augmentation anytime soon. But I do think there's sort of a natural timescale over which human augmentation occurs. If you look at drugs like antidepressants or Ritalin, if you look at non-invasive brain technologies, they start out helping people who have severe problems, but then as time passes, if they're found to be effective and not to have major side effects, their usage will spread. And I think that it's, in my estimate, maybe a 10 to 20 year period over which safety and efficacy can be vetted, and maybe a medical technology will start to veer into the realm of augmentation. Mm-hmm. But I think rushing it can fail. I've seen several companies where they tried to build a brain stimulator for some augmentation purpose, and nobody wanted to buy it because, mm-hmm. What do we know about the long-term effects of hacking your brain? Mm-hmm. The honest answer is we don't.
Okay. And the last question is something entrepreneurial. We talked about this, I think, yesterday or at breakfast. You mentioned, the technology, it's all feasible. We get enough scientists who can do these things. What's missing really is, if we found a company, a startup, is managerial talent, people who can really manage these companies. Because you founded a bunch of companies, but you can't find business people to run these things, right? Can you explain?
Yeah, it's a great question. I mean, I think in software, we have all these legends. You started Microsoft and Facebook and all these other companies where a college dropout will go and start the company and then they become billionaires, right? But in biomedicine, you don't see that happening. It takes wisdom and you have to deal with ethics and regulation and insurance. And I feel like it's a much more complex network of things that are going on in biomedicine. And so our biggest problem with our startups, we've spun out 4 companies from our group and we're planning 3 more, is actually finding people who can lead the way and to take products out of the lab and into the marketplace. So something I'm hoping to do is to figure out, how do we cultivate those leaders? How do we build, and train the biomedical leaders of the 21st century?
Thank you so much, Ed.
Thank you.
This was absolutely fantastic.